and 2 minutes will be devoted for questioning, discussions from the audience, which is desirable. So I request each and everyone to please stick to the time, 8 minutes, and then we'll really enjoy all of these presentations. The first one is Mr. Samrat Das.
A very good afternoon to one and all present here. I am Samrat Das. I will be presenting my talk on arsenic and smokeless tobacco-induced genotoxicity, sperm abnormality, and oxidative stress depends upon the nutrition status of mice. Before starting my talk, I would like to thank the chairpersons of these scientific sessions and all the organizing members of the EMSI 2017 for giving me an opportunity to speak in this prestigious EMSI conference 2017. Thank you one and all. So. Coming to the background of the study, arsenic, as we all know, is a naturally occurring metalloid. It is a well-known water contaminant which causes a wide range of serious adverse health effects, including cancer. Arsenic contamination of drinking water is a public health problem in Southeast Asia. And metabolism of arsenic includes series of reduction and oxidation methylation reactions that occur in liver to form monomethyl arsenic acid and dimethyl arsenic acid. Now, studies have shown that groundwater in many northeastern states, including Assam, has been contaminated with the arsenic. Recently, high arsenic concentration has also been detected in the groundwater of Barakvili region of Assam. And coming to the smokeless tobacco part, I'm working on a very unique smokeless tobacco, that is Sadagura. It is one of the most common type of tobacco chewing habits that is practiced and consumed indiscriminately in the population from southern Assam. And a paper by Koser et al. In 2009 in Mutagen research has clearly shown that the micronucleus and other nuclear abnormalities do occur among the chewers. So the Sadagura has the potential to cause genotoxicity in humans. Recently, I have found that arsenic and Sadagura have the potential to cause genotoxicity, male germ cell toxicity, as well as oxidative stress in mice test system. Coming to the arsenic transmethylation reaction, the detoxification mechanism of the arsenic actually involves S-adenosyl methionine, SAM, and it has been shown to be the primary methyl donor. Methylation reactions has been considered to be a detoxification step with respect to arsenic-mediated agotoxicity and teratogenicity. And also it has been seen that diets which are deficient in protein and methionine are poor sources of methyl donor groups. It has been found that dietary methyl group deficiency leads to a decreased SAM levels, and studies have suggested that reduced availability of free thiol groups, which predominantly occur with low dietary protein intake, might result in the enhanced expression of toxicity and altered arsenic metabolism. Hence, in view of the above, the present study was undertaken to test the effect of protein-rich diet towards mitigation of arsenic and Sadagura-induced genotoxicity, sperm morphology impairment, and oxidative stress over long-term exposure in mice test system for 90 days. This is a Mattel's method. Swiss albino mice were divided into 12 groups based on the protein content present in the diet, high protein, optimum protein, and low protein. The protein content was is based on the percentage of casein present. So I have followed the diet composition as followed by Chino et al. 2005. This is a table of the study design study groups for the high protein diets. There were 4 groups as control: sodium arsenite, Sadagura extract, and combination of sodium arsenite and Sadagura extract. For sodium arsenite, the dose was 2 mg per kg body weight per day. For Sadagura extract, it was 5 gram per kg body weight per day, and the combined dose was this. Similarly, for optimum protein series, also the same, and the low protein also the same. So total, there are 12 groups which I have the experiment has been done. After the chronic exposure of 90 days, genotoxicity assessment in femur bone marrow cells was performed. Micronucleus and comet assay was done. Germ cell toxicity parameters of sperm and abnormality assay and epithelial sperm count was done. Oxidative parameters in hepatic, renal, and small detached tissue was performed. Lipid peroxidation, GHS, and SOD, and histological examination by HE staining was done in liver and kidney tissue. I will not go in detail to the various protocols because as a shortage of time, I will just skip to the result section. This is the photographs of the micronucleus containing erythrocytes. So in the micronucleus incidence, as you can see, I observed statistically higher percentage of micronate content PC in all the test groups in low protein diet series. These four. These had comparatively higher incidence of MNPCs as compared to the optimum as well as high protein group. So next, coming to the cell cytotoxicity. PCNC ratio, lowest PCNC ratio was observed in case of the sodium arsenate plus Sadagura low protein group, while the highest ratio was found in case of the control high protein. So it gives an indication that high protein groups had comparatively higher PCNC ratio. So high protein might play a critical role in reducing the cytotoxicity as observed in the low proteins. So coming to the to verify the results as observed in the micronucleus assay, comet assay was performed, single cell gel electrophoresis assay. So this is a fold chain histogram of the head DNA, and lowest head DNA fold change was observed in case of this sodium arsenate plus Sadagura low protein group, while the combined groups while the combined groups in optimum protein and high protein showed comparatively higher fold change. And this is the tail DNA. I observed that sodium arsenide and Sadagura in low protein group caused the highest DNA damage, which was statistically higher as compared to its control lower protein. And also quantitative estimate showed significant increase in the percentage of the tail DNA in the sodium arsenide plus Sadagura low protein, and also in various other groups it was significantly different was observed. Similarly, in olive tail moment, the highest OTM fold change was observed in case of the sodium arsenide Sadagura low protein group. This is a damage index. Damage index is based on the first of all classifying all the cells based on their the extent of the tail, how much it has migrated, and again putting into a formula as described by Supriya et al. 2015. I have calculated damage index, and this is in which I have found that the damage index of sodium arsenide plus Sadagura high protein group, this group was significantly lower as compared to the sodium arsenide plus Sadagura optimum, and also the sodium arsenide plus SG low protein group. So this is a histogram showing the distribution of the cells. Comparatively lower damage cells were observed in case of the high protein, and sodium arsenide, Sadagura, and sodium arsenide plus Sadagura groups of low protein. This one, low protein groups had higher incidence of C4 cells showing extensive DNA damage. Again, this C0 to C1, this is based on the what is the tail length. For example, C0 is 0 mm and such and such. So C4 is equal to or greater than 20 mm length the DNA. So these are the results of the sperm head abnormality assay. I observed a higher incidence of sperm head abnormalities in case of the low protein series as compared to their optimum as well as high protein. So this is the sperm count. Yes. This is the sperm count results. And the results have suggested that high protein has protected against sodium arsenide as Johnson toxicity. Coming to lipid peroxidation of the MDA, there was statistically lower observation of MDA levels in hepatic HP as compared to the optimum combined group. Similarly, similar trend was observed in kidney as well as in small intestine MDA levels. GSH, significantly high levels of GSH in sodium arsenate high protein and optimum protein, and versus sodium arsenate low protein was observed. Similar trend was observed in as well as kidney GSH and also SIGSH. SOD, which plays a critical role in the dismutation of the dangerous chemicals. So lower levels of SOD was observed in low protein diet series in liver samples. Also in kidney samples, statistically higher SOD levels were observed in the high protein diet series. Coming to the SISOD, lowest SOD activity was observed in case of the sodium arsenate plus Sadagura low protein group. This is the histological results. The sodium arsenate high protein showed the cells with clear cytoplasm. Here it is clear cytoplasm. It's not clear, but see a clear cytoplasm suggesting cell death. Here the infiltrating cells are present in case of optimum protein, and here is the bigger cells, a karyomegaly cells showing extensive DNA synthesis. In Sadagura, there was disruption of the architecture, and here again in optimum protein Sadagura, there is disruption architecture as well as infiltrating cells. Here is a classical case of the metaplasia condition due to the tissue change in tissue parameter. In combined group high protein, there was incidence of intestation infiltration, and cells with clear cytoplasm here with suggesting cell death, and here again cell with clear cytoplasm suggesting cell death. And coming to the next picture, this is the renal histology slides. Here infiltration, here again infiltration, here is metaplasia yeah, only 2 slides, ma'am. This is the metaplastic conditions. So further I have done GCMS analysis for Sadagura extract, and including nicotine, there was various other harmful compounds which I have observed to be present. These are my references, and thank you so much.
Now this is open for discussion. Few questions, 1 or 2.
In addition to variable protein content, whether all 3 animal groups received isocaloric diet, because otherwise that will be an additional variable.
So that's why, sir, for all these series there was control. There was control group was there. So if there is some variation due to the diet.
Excuse me.
Yes, sir.
This giving high protein, you want to say that same level goes to high protein diet?
I have not checked the same levels. I've just looked into the general.
Theoretically, can it increase the same level with giving high protein diet?
High protein diet will obviously have high concentration of the thiol groups, and
That is okay. No objection with that, but SM level I will.
SM levels, it will help in the methylation reactions. So it will lead to the higher excretion rate of the arsenic from the body. So it has potential to show protection against the observed arsenic and Sadagura toxicity. Yes, ma'am.
Excuse.
Yes.
Thank you. Very nice presentation.
Thank you, ma'am.
I just want to know that methylation, you are telling that it is the cause of detoxification. But recent theory also says that methylation is also a toxification of especially the DNA is carcinogen.
Mhm.
The formation of the trivalent methylated forms are very carcinogenic forms. So these are more toxic. Any comments on that part?
Ma'am, there are research going on in various ATP binding cassette proteins, like MRP, in the Canada, Professor Alan Leslie lab. They have found that the arsenic will combine with the GSS to form various which conjugates, arsenic conjugates, which in turn gets eliminated via the urine. And they have done in HEK cell lines, and studies are going on. So methylation will obviously play critical role in the detoxification. Why I'm telling that? Because if there is by the results suggest, if there is decrease in the protein content, so there will be obviously there will be less thiol groups present, and less thiol groups presence means against the conjugate formation with the arsenic and thiol groups will not take place. So this might in turn delay in the excretion of the arsenic from the body. So yeah.
Okay. Just one small question from.
Just one comment that during this methylation part,
Yes, ma'am.
you should, because methylation is very important regarding arsenic metabolism.
Yes,
You should also look into the methylation status, into the epigenetic regulation, because arsenic is well known for hypomethylation, lower DNA hypomethylation, as well as promoted hypomethylation. So all these should be considered while you are trying to serve for thiol groups. So please, you must have a
Okay.
you should look into these points.
Okay, you can discuss
Thank
little
you.
further.
Thank you for research.
Dr.
Thank you so much.
Saraswati, just
Yeah.
last question.
Excuse me, a very good presentation, Das. And
Thank you.
here, I just want to know that you had presented, because I did a little bit of work in sodium arsenite with Professor A.T. Natarajan. So I just wanted to know, you have not taken up chromosomal aberration essay, but
CF.
you have taken up CBM. Any particular reason?
Which one? I've taken MN comment essay.
Yeah.
Yeah.
Or any particular reason why you did not take up CA?
Ma'am, since MN will be, it will be looking into the whether it's directly inhibiting the.
Okay, Dr. Das, I think you should discuss later on because
Thank you.
we are
once
running
again,
out of
everyone.
time. And I'm requesting again to the presenters to please keep time of 8 minutes. The next speaker will be Ms. Ira Seth. Ms. Seth, please stick to 8 minutes only, please.
Good afternoon, one and all. I'm here to present my work entitled Ameliorative Potential of Acyl Marmelos on cadmium-induced oxidative stress and leading to neural histogenotoxic alterations in murine model. In the present study, attempt was made to understand is there any correlation between cadmium-induced oxidative stress and the histogenotoxic alterations which were induced by the subacute exposure to cadmium. So cadmium is a heavy metal. It is a pervasive essential heavy metal and is considered as a pollutant ranked 7th in top 20 hazardous priority list. It is a unique heavy metal because of its low excretion rate and high rate of accumulation in the body. It has a half-life of 15 to 20 years. So exposure to cadmium has been broadly classified into two categories, that is occupational and non-occupational. So the workers working in smelters, smelters and other factories which use cadmium and produce it are directly exposed and so are considered as occupational exposure. The non-occupational exposure is mainly through ingestion and inhalation by using the products which use cadmium indirectly, such as rechargeable batteries, cigarettes, pigment stabilizers, cosmetics, jewelry, and etc. Cadmium mostly affects every organ system of the body, may it be nervous system, liver, reproductive system, kidney, skeletal system. It has been considered as a group B1 carcinogen. And in the nervous system, it causes hemorrhages and neuropathology. How cadmium enters into the neurons? It enters via the calcium-mediated channels, and once inside the cells, it produces free radicals by indirectly interacting with the critical cellular sites. These free radicals in turn increase the levels of lipid peroxidation, causing disintegration of the biomembranes and the mitochondrial membranes. They also cause decrease in the endogenous antioxidant compounds, enzymes, such as catalase, superoxide dismutase, peroxidases. So not good medications are available for the metal poisoning. So, and they are all burdened with side effects. So we choose a natural antioxidant to cure the ill effects. The plant chosen here is Acyl marmelos, also known as bel patra. It belongs to family Rutiaceae. It has been extensively described in the Vedic literature for the treatment of various diseases. The leaf extracts of acyl are known to acquire several bioactive components such as acyline, quercetin, myricetin. They are alkaloids and flavonoids respectively. Coming to the phytochemical analysis, the preliminary qualitative estimation of ethanolic extract of acyl revealed the presence of alkaloids, carbohydrates, phenols, tannins, flavonoids, and terpenoids. These phytochemicals provide acyl with its pharmacological properties. Acyl is also reported to possess several properties such as hepatoprotective, antioxidant, cytoprotective, antimicrobial, and many more. So the aim of the present study was to examine, and the rationale was to study the clustergenic effect of the cadmium in regard to the biochemical and the histogenotoxic alterations by investigating the oxidative stress markers, LPO and GSH, histopathological changes in the brain tissue, and the bone marrow chromosomal analysis, and the micronucleation in the bone marrow. Coming to the methodology, there were 6 groups. The experimental animals selected were adult Balsi mice. The control group, the cadmium group, was given 5 milligram per kg body weight of cadmium intraperitoneally for 5 days. The AGL group was given 500 milligram per kg body weight of AGL for 10 days. There were 2 treatment groups, pre-treated and post. The pre-treated was given AGL for the first 10 days and cadmium in the next 5 days. And the post-treated group was given cadmium for the first 5 days and then AGL for the next 10 days. The studies were biochemical, histopathological, and genotoxic. Coming to the results and discussion, biochemical studies. When cadmium was administered for 5 days, there was a statistically significant increase in the levels of lipid peroxidation. A 50% increase was observed, whereas in the AGL treated group, the values were to near normal values is that of the control. The pre-treated group had more values closer to the control group as compared to the post-treated group. There was a statistically significant decrease in the levels of GSH, and even here was found that the post-treated group were moderately effective as compared to the pre-treated groups. So the antioxidant enzymes become inactive by the direct binding of cadmium to the enzyme's active sites containing the sulfahedral groups. After cadmium intoxication, there is disintegration of the biomembranes, which lead to cellular leakage of the endogenous antioxidant enzymes. The presence of several active phytoconstituents are the major possible reason for the restoration of the levels of these enzymes by AGL. Coming to histopathological study, these are the light micrographs. Coming to the histopathological study, these are the histomicrographs of the brain. The control group, there are 3 regions studied: cerebellum, striatum, and the cerebrum. The control group reveals a well-defined molecular and granular layer, and at the junction of them are present Purkinje cells. In the striatum, the pink background is of the neuropil, which is a mat of glial cell processes and the neuronal cells. The cerebrum shows well-defined pyramidal cells and the fusiform cells in the neuropil. In the cadmium-treated group, after 5 days of exposure, there was a loss of Purkinje cells at the junction of the molecular and the granular layer. There was pycnosis in the nuclei at the striatum, and there was vacuolization and interstitial edema in the striatum. Hyperchromatization of the pyramidal cells in the cerebrum was observed with pericellular halos around them. There was also excessive vacuolization in the neuropil after cadmium exposure. Coming to the treated group, the pre-treated group revealed much normal histomicrographs as compared to the cadmium, as compared to the post-treated group, as we can see the Purkinje cells are much more formed and lined in the pre-treated group as compared to the post-treated group. And the post-treated group still more vacuolization and interstitial edema is seen. The free radicals attack the polyunsaturated fatty acid cell membranes, leading to destabilization and disintegration of the membranes. Brain tissue is highly susceptible to oxidative damage due to its high utilization of oxygen. So the histopathological alterations are moderated by administration of AGL because of the presence of these active phytoconstituents. Coming to the genotoxic studies, chromosomal aberrations were studied and they were divided into 4 groups: the structural, exchange, and the physiological and numerical. The total number of aberration in the control groups were 56%. These are the photomicrographs of the metaphasic plates. The cadmium group shows ring formations, condensed chromosome, sticky chromosomes, centromeric fusion, multiple chromatid breaks. a very few chromosomal aberrations were observed in the treated groups. And at last, coming to the micronucleation, the control in the cadmium group, there was a 16-fold increase in the level of micronucleation as compared to the control. The post-treated group did not give a better result. There was a 12% increase in the micronucleation as compared to the control. So the cadmium affects the genome stability by oxidative stress inhibition of the DNA repair mechanism, and these effects play an important role in increasing chromosomal structural aberrations and micronuclear formation. Coming to the summary and conclusion, the present study demonstrated the modulatory effect of AGL against cadmium-induced oxidative stress at the cellular and subcellular level. So it is recommended that awareness should be focused on AGL and its bioactive phytoconstituents as an antioxidant in dietary supplements. Considering the mild degenerative changes still observed in some cases in this work, further intensive experimental and clinical study may be required to adjust the dose, time, and effective active phytoconstituents of AGL. This is the bibliography. Thank you.
Okay, now we have crossed time, so there will not be any discussion. Thank you so much for a nice presentation. So the next participant is Miss Priyanka Prasad.
A very good afternoon to all respected judges, respected chairpersons, eminent scientists, and my dear friends. Arsenic, as we all know, is a global public health concern and is widely prevalent in 9 out of 19 districts of West Bengal and is afflicting 20 million people residing in these areas and solely dependent on these aquifers. World Health Organization has recommended the safe limit for this class 1 carcinogen at 10 microgram per liter, but Bureau of Indian Standards concedes it at 50 microgram per liter wherever alternative sources are unavailable. Thus, we see that there is a huge difference between these two organizations' recommended legally enforceable limit. Here we see hallmarks of arsenic toxicity, and moving on. we already know that around 48% of groundwater in West Bengal is contaminated with arsenic in a range of 11 to 50 microgram per liter, and people in these areas that is around 90% of the population is dependent on these aquifers. Since it is a low arsenic content region and since the concentration is low, hence these people don't show any don't show much symptoms of arsenicosis, but they may be subclinically affected, and this is what was our lookout. We were interested to investigate the effect of this low level on sputum cytological alterations as well as inflammation. Sputum was selected because it is surrogate of lung, and which in turn is a target organ of arsenic. The areas selected for this study were two districts from West Bengal. District was Noida and North 24 Parganas, where Noida had arsenic content below 10 microgram per liter and was a control area, whereas though the areas in 24 Parganas North that had arsenic content in a range of 11 to 50 microgram per liter was our exposed area. And this study was approved by Institutional Ethical Committee. Only those women samples were included for the participants were included for this study who did not show any symptoms of arsenicosis and had been residing in those areas for a minimum of 10 years. Samples collected were groundwater, nails, sputum, and blood. And a detailed demographic study was carried out to establish the fact that these controlled and exposed women were comparable for age, schooling, their household activities, as well as their lifestyle. While when we analyzed arsenic content, we found that groundwater arsenic in exposed area had around 29 microgram per liter, while the control area had arsenic as a mean of 2.3 microgram per liter, which is quite significant in comparison to the exposed area. And a positive correlation was obtained between arsenic content in nail with that in groundwater, which upholds the fact that these participants are chronically exposed to arsenic content. Next was our objective, that was sputum cytological alteration assessment, where we found that low-level exposed women had increased frequency of metaplastic epithelial cells, had enhancement in multinucleated epithelial cells. Goblet cell hyperplasia was more in case of these women, as well as mucosal deposits was found to increase. Besides, presence of celiocytothoria and coilocytes, which are associated with viral infection, was also found to be more prevalent in exposed area as compared to the control individuals. Here the next was to look for inflammatory cells, where we found there was an upsurge in the number of eosinophils, lymphocytes, neutrophils, and alveolar macrophages in the exposed women as compared to the control. Besides, when we went to look for alveolar macrophages, we found that around 16% of the total alveolar macrophage was small sputum macrophage, which had this thing, which were functionally immature. Elastase activity of neutrophils was found in all the cells, but where in case of control samples, 58% of the neutrophils had highest grade of activity. Arsenic exposed women had shown highest grade of activity in 81.7% of the neutrophil cells. Similarly, in case of alveolar macrophage, we found that around 25% of the cells had alveolar activity as compared to that of the 10%, and of them 10% in controls, and of them 5.5% of the alveolar macrophage were shown with highest activity as compared to 0.2% in the control group. Gelatinous activity was also increased in arsenic exposed women. And next was to look for the sensitive pro-inflammatory receptors, where we found that CD16 and CD14 both had enhanced it in expression in case of arsenic exposed women. Next was to look for the inflammatory signaling associated with inflammation, since it was one of our objectives. Here we found that TNF alpha, which is downstream to CD14, was also enhanced in expression both in airways as well as in plasma. Similarly, the downstream molecule of TNF alpha, which activates actually NF-kappa B, downstream of TNF alpha, NF-kappa B was found to be upregulated in airways, and this is associated with goblet cell metaplasia, as well as it controls the pro-inflammatory cytokines transcription, which are IL-6, IL-8, and IL-12. Here we found that IL-6, IL-8, and IL-12 were also upregulated in plasma samples. Besides, we also found that AKT, which may be associated with goblet cell hyperplasia, which was one of our findings, was also upregulated. The limitations to this study was that since it's a cross-sectional study, inference in causal direction may be limited. Arsenic input from food chain could not be analyzed, and total input of arsenic of an individual during the lifetime was only speculative. But besides confounding factors from all the from all other toxic insults, for example, agricultural pesticides could not be totally removed by multivariate logistic regression analysis. But since our sample size was large enough, we could summarize that chronic low-level arsenic exposure may be associated with cytological alteration that result in accumulation of inflammatory cells, is one of the reasons for goblet cell hyperplasia, metaplasia, and is one of the reasons for celiocytothoria and coilocytes, as well as for multinucleated epithelial cells presence. Here we can say that presence of multinucleated epithelial cells signify that there may be cell cycle deregulation. Likewise, presence of goblet cell hyperplasia as well as metaplasia may provide a direction towards lung tissue injury, and mucosal secretion, as we see in enhanced mucosal deposits, as we see in lung, is associated with goblet cell hyperplasia and may be indicative of recurrent infection. All these changes that we are seeing in lungs may induce inflammation, which is associated with severe diseases, including chronic diseases like malignancy. Thank you.
We have only 1 minute, so we'll have 1 or 2 very short questions, please. If there are no questions, then I thank Ms. Chopra for a very good presentation. Next presenter will be Ms. Krishna Bhuiya.
A very good afternoon to everyone. I'm okay. A very good afternoon to everyone. Myself, Krishna Bhuiya. My presentation is on the topic that is phenytoin induces oxidative stress, DNA damage, and neurological imbalance on the tadpoles of the Indian skittering frog, that is Euphlyctis cynophlytis. First of all, I would like to. That is not going. First of all, I would like to draw your attention towards a group of persistent pollutant of concern, which is produced and released to the environment by the incomplete combustion of fossil fuels and all other organic substances, that is the polycyclic aromatic hydrocarbons, which is commonly known as PAH. There are more than 100 PAH compounds found in the environment, but I have chosen phenytoin as my chemical of interest as because it is the most abundant PAH found in the environment, especially in the aquatic environment. The phenytoin is also included in a list of 16 priority pollutants by the US Environmental Protection Agency. I have studied the effect of this phenytoin on my model organism, that is the enduron amphibian tadpole, more specifically the tadpoles of Euphlyctis cynophlytis. Now the methods and methods. My methods and methods starts with the collection of tadpoles and then acclimatization in the laboratory condition, and then this identification and separation of the desired stage of the tadpoles according to the Gostner 1960. This schematic diagram represents how I determine the LC50 values for phenytoin. After calculating the LC50, I have used 5% and 10% of the phenantrin 96-hour LCVC concentration for the study of my oxidative stress parameters, as well as the neurotoxic potential, to study the neurotoxic potential, and the minimal concentration, that is the 1% of the 96-hour LC50 I'm using it for the to study the genotoxic potential, that is by Comet assay. Okay, I have studied for oxidative parameters. I have studied the SOD activity, JSS, and lipid peroxidation. For genotoxicity assessment, I have studied. I've done the Comet assay, and for neurotoxicity assessment, I have done the acetylcholine estrase assay. So these are the detailed procedures I have followed during my experimental procedures. Now the results and discussion. This table shows the LC50 values of the phenantrin when it exposed to the euplictic sinuplictic tadpoles during different time points. As you can see that in the 24-hour LC50 values for phenantrin is 16.52, whereas for 48 hours it is 15.29, and for 72 hours it is 13.69, as well as for 96 hours it is 12.28 mg per liter per concentration. Linear regression analysis of the LC50 concentration of phenantrin over the time periods of 24 to 96 hours, as we find that with increasing time duration, the LC50 concentration is going down. Now, this slide shows the sulfur oxide dismutase activity as well as JSS levels when we expose to the euplictic sinuplictic tadpoles. So I found that the phenantrin causes excessive induction in the superoxide dismutation, superoxide SOD levels as well as JS and JSH. The Lustak 2010 proposed a theoretical framework in which that when a subject is exposed to an oxidant, that what happens, the host body in a chronic situation, what happens if the host body overproduces or upregulate the antioxidant system as an adaptive response to attenuate this ROH generations. So in my generation, so that the ROS level slows down at the later stages. In my study, I found that increased SOD and GSH could be an adaptive measure by the tadpoles to compensate the ROS generation following phenantrin exposure. But interestingly, I have found that the data on lipid peroxidation, it shows the lipid peroxidation shows that clearly shows that although even though the tadpole body is even though the tadpole body is activated the adaptive mechanism, but still the adaptive mechanism to reach the ROS formation, but still the oxidative damage still occurs in the inflicted and inflicted tadpoles which is evidenced by the increased amount of MDA formations. Exposure of total tadpoles to a nominal concentration of phenantrin for 48 hours resulted in DNA damage, as evidenced by migration of DNA fragments in the gels forming comet-like structure. This photomicrograph is showing the different types of damaged comet's nucleus with damaged nucleus. A quantitative distribution of cells with damaged DNA is demonstrated in a figure A, and from that distribution, I have also calculated the damage index, which is very much significantly increased as that of the control. The quantitative parameters such as tail DNA, olive tail moment, and tail extent moment is also shows the similar trend as that of the damage index. That means in case of the treated samples, it shows a higher amount than that of the control. But the results is very contrasted with the head DNA, because in head DNA, in treated groups, it lowers than that of the control, which proves the DNA damage. The DNA damage that caused in my study, it may be due to the direct interaction between the PAH compounds and to the DNA, which may lead to the DNA single strand break, DNA double strand break, DNA adduct formation, etc. And also it may be the ROS generation. What I found is maybe indirectly affecting the DNA damage system. Now, it is histogram showing the acetylcholine esterase activity of the afflicted synafflicted stat pole. We can easily see that when phenanthrene exposed to the afflicted synafflicted stat poles, the acetylcholine esterase activity gradually increased significantly. So as we all know that acetylcholine esterase is a primary cholinesterase found cholinesterase that catalyzes the acetylcholine neurotransmitters. So the increased acetylcholine esterase, which leads to the diminishing amount of acetylcholine, which may cause the reduction in the cholinergic system neurotransmitter efficiency. And but still the increased acetylcholine activity, the mechanism behind this increased acetylcholine activity is still unknown. So overall, that overall findings suggest that phenanthrene is a quite toxic chemical which can affect which can cause many sublethal effects in the my model organism, that is phenanthrene. Oh, sorry, that is the afflicted synafflicted that pulls by enhancing the oxidative stress parameters by leading to DNA damage and induce acetylcholine esterase formation. That's all. These are references. Thank you very much.
Thank you, Ms. Maya, for keeping time. So this is open for discussion. Any question? Please stand up. Okay. So thank you again.
Thank you very much.
Okay. Our next speaker will be Mr. Siddhama Tilagul Kumbar.
Okay. Good afternoon, all. I'm here to present my research work, which is entitled Organophosphate Pesticides Induced Oxidative Stress Mediated DNA Damage in Human Lymphocytes. As we all know, cancer is one of the devastating diseases of the present world. So according to the recent report, there are around 60 lakhs deaths per year, and it is second and third leading cause of the death in developed and developing world. And World Health Organization estimates that by 2020, there will be around 100 lakhs deaths per year. If you look into the cancer scenario of India, at any time, there are 30 cases and 11 lakh new cases are being registered in hospital. And every year, 6 lakh people die. And if we look into the epidemiology, leukemia is one of the major cancers that is occurring. And leukemia is the most common form of cancer in children, that is called childhood leukemia. And it covers 41% of the malignancies that occur in children. And among leukemia, acute lymphoblastic or lymphocytic leukemia covers 77%. And still, cases for this leukemia, mainly cancer and childhood leukemia, remains enigmatic. One epidemiological study states that this leukemia or childhood leukemia occurrence is more in the agricultural related areas. So based on that, they found that people related with farmer families related with these agricultural areas where these pesticides are sprayed, there is increased incidence of cancer, especially childhood leukemia. So based on that, they hypothesized that pesticides might play a major role in this incidence. So via parental exposure prior to conception, in utero, and direct exposure during childhood. And India is the second largest producer of pesticides in Asia, with around 90,000 metric tons of pesticides produced annually. So among them, organophosphate pesticides are used more, nearly 40%. Among them, malathion and perothianas are used extensively. So based on these epidemiological studies, we hypothesize that exposure of human lymphocytes to organophosphate pesticides leads to reactive oxygen species mediated DNA mediated oxidative stress and chronic DNA damage that may influence the initiation and promotion of leukemia. So these are the objectives of my study. And first is to analyze organophosphate pesticides induced oxidative stress in human lymphocytes and to evaluate the DNA damage occurred in human lymphocytes after exposure to these pesticides. So we selected malathion and parathion. First, we isolated the lymphocytes by healthy donor blood after getting the permission from University Ethical Committee. Then we initiated the culture by adding mitogen like phytomyoglutamine. And to know the inhibitor concentration of these malathion and parathion, we did MTT assay. After that, to see the oxidative stress parameter, we saw reactive oxygen species generation, and to see lipid peroxidation, we measured the level of malondialdehyde. And as an oxidant marker, we saw the superoxide dismutase activity, catalase activity, and we did the total glutathione content, measured the total glutathione content. After these biochemical assays, to assess the DNA damage occurred, we did COMET assay. So we found that malathion, inhibitor concentration of malathion is 218 microgram per ml, and parathion is around 148 microgram per ml. And based on this, we fixed the sublethal concentration of malathion and parathion. In every experiment, we kept N-acetylcysteine as positive control. So these are the results. What we got? There is increased reactive oxygen species production in the exposed group when compared to control, N-acetylcysteine treated and N-acetylcysteine treated plus pesticide treated group. So we can see there is dose dependent increase in the production of reactive oxygen species. After this, a second oxidative stress marker, that is malondialdehyde content, which occurs as a means of lipid peroxidation. Here also we can see treated groups show increased MDA level. And as an antioxidant marker, superoxide dismutase assay is decreasing. This may be due to the increase in the oxidative stress of the cell, which may be produced because of the exposure to these pesticides. And second antioxidant marker, that is catalase. Here also activity is lesser activity when compared to control and N-acetylcysteine treated and N-acetylcysteine treated plus pesticide treated group. So another antioxidant level marker, antioxidant marker, that is total glutathione content. Here also you can see reduced total glutathione content of the cell. So if we look at the graph, we can clearly make out that parathion is slightly more toxic than the malathion. After this biochemical assay, we did a COMET assay. This is a representative images of treated group. Group 1, group 2, group 3, and group 4 are the treated groups. So here we can see tail, DNA tail, which is produced because of this DNA damage, DNA strand breaks. This is the tail length. Here we can see tail length is increasing in the treated group than the control. So based on this, I can summarize that lymphocytes exposed to these organophosphate pesticides show increased level of reactive oxygen species production. These reactive oxygen species production will cause oxidative stress in the cell, and oxidative stress will cause the decrease in the antioxidant level of the cell. This decrease in the antioxidant makeup of the cell will alter the DNA repair capacity of the cell, and free radicals formed will react with the DNA and to form DNA oxidative lesions, and finally to cause cellular toxicity. And as a result, DNA double strand breaks or chromosomal aberrations can be seen. So based on this, I can tell that oxidative stress plays an important role in the organophosphate pesticides induced cellular cytotoxicity, genotoxicity in the initiation or onset of the childhood leukemia. Thank you.
Okay, questions? Who can? Any questions?
I think
while I can understand that these pesticides, they are affecting the blood lymphocytes and bringing about so many damage, I don't know why you want to link it with the leukemias and all these things. Can you tell me the connection between the two?
Why I want to means I aiming to link to leukemia.
Yes.
Because leukemia is childhood leukemia especially is one of the major diseases can be found in agricultural sectors. Children with below age 5 to 6 age, they are dying. There is no cure.
But where is this epidemiological study actually? You have got the details of this epidemiological study that they have proved that exposure to organophosphor, this thing, pesticides, they increase the incidence of childhood. What is the reference for this thing?
Actually, I have not. Actually, I have not put.
To give a reference for that too. Because
to be in time, I have not put, but I have got papers. Not only in India and Spain, there also it was performed in many regions of the world.
No, have they established that there is a causal relationship between the exposure to these pesticides and increase in the incidence of leukemia, or it is only a observation?
We are checking gene expression parameters also, sir, further after this.
Any more questions? Still we have time. Okay, thank you so much for a nice presentation.
I would like to thank organizers and Environmental Mutagen Society of India and my supervisor for giving me an opportunity to present my research work. Thank you one and all for your kind attention.
Okay. The next speaker is Dr. Vinay Jain.
A very good afternoon to everyone. The title of my talk is Evidence of Efficient Repair of DNA Damage and Involvement of DNA Repair Genes in Individuals from High-Level Natural Radiation Areas of Kerala Coast, India. We all are exposed to low levels of radiation through various sources, be it natural or man-made, and the biological effects of low-dose radiations in human population is not well known. And so it is of great interest to investigate the biological effects of low-level radiation in human population. High-level natural radiation areas give us an opportunity to study the effect of low-level radiation directly on human population. We have a HNR area in a Kerala coast, which is extending a strip of 55 kilometers, where the most unique feature of this area is that here the background dose rates varies from 1 milligram per year to 45 milligram per year, which is ideal to carry out background dose-related dose response studies. Various studies have been carried out in this area, but there is a need to implement more sensitive and high throughput markers to find out any molecular changes are there. So we have introduced a sensitive double-strand break marker gamma H2X to analyze the spontaneous frequency of double-strand breaks in that population. The study was carried out in 91 individuals, where we did not observe any background dose-related increase in the frequency of double-strand breaks. We divided these individuals into 3 different dose groups. The control area group, which received less than 1.5 milligram per year, low dose group where individuals from 1.51 to 5 milligram were there, and more than 5 milligram was characterized as HDG individuals, high dose group individuals. And as can be seen in the graph, we observed a marginal reduction in the DSB frequency in the HDG individuals as compared to the control areas. We we studied the relationship between the age and the foci frequency, as it is known that with increase in age, DNA damage increases. Interestingly, we observed in HNR individual, there was significant increase in the frequency of DSBs with age. However, in HNR individuals, as we can see, there is no increase in the frequency of DSBs with respect to age. It is an interesting result in that sense. We further carried out the induction and repair analysis of DNA DSBs by giving the challenging doses. As we can see, we observed at 0.25 gram of challenging dose, there was a significantly reduced induction of DSBs in the high dose group individuals HNRA as compared to control individuals. We carried out the repair kinetics of DSBs by at different post-irradiation time points. And we observed at both the at 0.25 gray as well as 2 gray doses, we observed significantly faster repair of DSBs in the high dose group of HNRA population as compared to the normal individuals at 4-hour and 6-hour time point. We wanted to study the role of DNA DSB repair genes in this faster repair efficiency observed. We studied the transcription of non-homologous end joining repair pathway genes in at basal level, as well as after giving a challenging dose of 2 gray at 4-hour time point. And we observed that at baseline level, Cu70 and Cu80 genes showed upregulation. And after giving a challenging dose of 2 gray, we observed important genes like XRCC4, Artemis, DCR1C, and Cu80 showed significant increase in expression, which suggests that NHEJ repair genes may be playing a possible role in the in vivo adaptation of the faster repair of DSBs observed in that population. Taking cue from this, we carried out global transcriptome profile analysis in that population. We studied 4 different dose groups, control, LDG, and 2 different dose groups of high dose group of HLNRA. This is our approach. We carried out blood from those individuals. We made multiple aliquots. One aliquot was taken for microarray analysis, and another aliquot was taken for validation using real-time PCR. And we further validated using a different set of 24 individuals to find out whether our results are consistent within the population or not. This is our results. We observed a background, as you can see, these are the dose groups, a background dose dependent increase in the number of differentially expressed genes in HLNRA population as compared to the control areas. We find out the common genes between different dose groups, and we observed around 82 genes to be common in different dose groups. And these were the functions played by these genes. They were involved in transcription regulation, mRNA processing, and few were involved in the metabolic processes. Since we observed very large number of genes to be differentially expressed at higher dose groups, we carried out a detailed gene ontology analysis to find out the function of these genes. And we observed most of the genes were involved in DNA repair, apoptosis, immune response, chromatin modification, cell cycle regulation, and signaling pathways. And we wanted to see what are these genes. We observed as they are the important genes involved in DNA damage response and repair, which were differentially expressed in our population. The other set of the genes, DNA repair genes, which are differentially expressed in that population. As you can see, all the important DNA repair pathways are represented in this, be it DSBs, base excision, NER, and other pathways. We carried out the pathway analysis to find out what are the pathways which are upregulated there, and we observed that important pathways like MAP kinase pathway and P53 pathway, which are known to be stress-induced and DNA damage response pathways, are overrepresented in that population. So in a nutshell, we observed all the important processes of DNA damage response to be active there. DNA repair, these genes were involved there. Cell cycle regulation, apoptosis, chromatin modification as important transcription factors involved in the P53 as well as MAP kinase pathways. We further looked for the genes which showed dose-responsive increases within those groups, and we observed these genes showed dose-related increase in their expression level from group 2, 3, and 4. And these are important genes, and they may be the possible chronic low-dose deletion signatures which in that population and can be further validated. And validation is very important aspect of any microarray study. So we carried out extensive validation in the samples on which microarray data was carried out. And we conducted a fresh samples of 24 individuals to find the consistency of our results. We used probe-based real-time PCR approach, and we observed a very comparable results in all the samples collected from that area, which gives the consistency to our results. And this is just a schematic representation from our side. We propose that because of the low dose addition, there is a change in the chromatin structure. A lot of chromatin modification genes are there. So maybe some epigenetic regulation is happening there. Important signaling pathways like P53 and MAP kinase are activated there, which leads to the increase in transcription of DNA repair genes. And if the DNA repair is not happening, apoptosis is another important pathway which removes the cells which are not being repaired, so that the mutation is not carried forward. And to summarize my results, we observed no background dose dependent increase in the DSB frequencies in HNRA. However, we observed a marginal reduction in DSB frequencies. Significant repair was observed in HNRA. Significantly efficient repair was observed in HNRA individuals as compared to NRA. Dose dependent increase in the number of differentially expressed genes was observed. Important pathways like DNA repair, cell cycle regulation, immune response were high in individuals of higher background dose groups. MAP kinase and P53 signaling pathways are active in that population, and we observe the dose responsive genes, which can be important chronic low-dose radiation exposure signatures. Thank you.
Thank you very much, Dr. Jain. Now this paper is open for discussion. Any questions, please? Okay, if there are no questions, thank you again. Oh, there is, okay.
You said something challenging studies. What do you mean by challenging study?
So what we do there is we have a population there which are exposed to background radiations to study that adaptive response studies where we consider the elevated level of background radiation they are receiving is a priming dose, and we challenge the cells PBMCs from that population with a challenging dose. Cells PBMCs we challenge with a challenging dose.
Any more questions? Okay. Thank you, Dr. Jain. Now, next speaker will be Ms. Monica Shirley Mani.
Good afternoon, everyone. I'm Monica, and I'm here to present a part of the work entitled Effect of Allied VDR and HFE Polymorphisms on Blood Lead Level Variation in Lead-Exposed Workers. We are all aware of a heavy metal called lead found in the periodic table, but how far are we aware that the water we drink or the food that we eat contain traces of lead that can accumulate or become a part of us in the course of our lifetime? So lead plays a very important role in our everyday life, and it is a potent non-biodegradable environmental public health toxin, and this constitutes about 0.6% of the global burden of diseases. Every individual vary in their response to toxicity, and this depends on various endogenous factors like age, gender, lifestyle, ethnicity, socioeconomic status, and most importantly, the magnitude of exposure. In addition to these factors, there is another key that can influence the susceptibility to toxicity, and this is the genetic and the epigenetic makeup of an individual, which is currently an area that is gaining thrust in establishing the toxicogenomic or toxicoepigenomic profile of any toxic element or toxic substance for that matter. In nature, most of the sources of lead are divalent, exist in a divalent form, and due to this, lead can interact with other divalent essential metal ions that are involved in the metabolism, and some of them include calcium, magnesium, zinc, etc. On this basis, we selected 3 pathways that is known to be potent inhibitors of lead. That is 1, heme biosynthesis, 2, in calcium signaling, and in iron regulation. In the heme biosynthetic pathway, lead inhibits 3 important enzymes. 1 is the allied, that is aminolevulinate dehydratase, 2, copropopenogen oxidase, that is CPOX, and ferrochelatase, that is FETCH. But among the 3, lead shows a stronger affinity to allied compared to the CPOX and FETCH. Then in the calcium signaling, the vitamin D receptor play an important role in regulating the levels of lead, calcium in the bone as well as in the blood circulation. Now, in the presence of lead, the calcium binding protein conforms its affinity more towards lead than calcium. So, as a result of this, lead gets absorbed into the system and deposits in the bone. In the iron regulation, there is a protein called HFE. It goes by the name as hemochromatosis. This protein regulates the level of iron inside the cell. Now, There are several studies which show that mutations in the HFE gene can disrupt the regulation of iron levels inside the cell, resulting in the overloading or the uncontrollable shuttling of iron inside the cell. And since lead and iron share similar divalent character, lead can lead tends to enter inside the cell along with the iron, and this leads to toxicity. So based on these concepts, we selected polymorphisms in the ALAD gene, VDR, and HFE gene. So this study mainly aims to investigate the influence of the ALAD, VDR, and HFE polymorphisms on the blood lead levels of lead-exposed subjects. So for that, we first analyzed the prevalence of the selected polymorphisms among the exposed individuals, and then correlated the genotype with the phenotype, which in our case is represented in the form of blood lead levels. This is the study protocol. First, we collected the blood samples from individuals working in an occupation which involves lead. And after obtaining their informed consent, the lead levels were estimated using the ESA model 3010B lead analyzer. And from the whole blood, we extracted the genomic DNA and subjected it to PCR RFLP to identify the genotype of every individual. So here in the first graph on the left is representing the distribution of the blood lead levels of the individuals that we had screened. In our study, we screened around 272 individuals, out of which 159 are lead exposed workers, and the rest are 113 control subjects, considering the fact that their blood lead levels are less than 10 microgram per deciliter, that is the acceptable range, and that they are not involved in any lead based activities, and they reside in areas away from lead contamination sites. Now, in addition to blood lead levels, there is another sensitive indicator that can measure the lead exposure, and that is the ALAD activity. So we performed the ALAD activity assay in all the individuals, and we observed that the lead exposed individuals who had very higher blood lead levels showed low ALAD activity compared to the control subjects, as indicated here. And we further did a correlation analysis, and here too we observed that a significant negative correlation, indicating that with increase in the blood lead levels, the ALAD activity reduced. With this information in mind, we next wanted to see if the ALAD genotype has any influence in modifying the blood lead levels. So here you can see the distribution of the blood lead levels in the different ALAD genotypes according to the codominant and the dominant inheritance model. And here we observed that the individuals with the individuals heterozygous for the ALAD SNP GC showed higher median blood lead levels compared to the other genotypes. Similarly, in the dominant model with the combination of GC and the CC genotype, we see a higher median blood lead level and corresponding lower ALAD activity. From this, we can infer that the C allele in this ALAD SNP locus might have a contributing risk factor in increasing the blood the retention of lead in the blood and the corresponding reduced ALAD activity. Next, in VDR polymorphism, we selected 3 SNPs which are located adjacent to each other near the 3' UTR region. These 3 SNPs do not determine the vitamin D receptor structure polymorphism, but there are studies which hypothesize that they have some effects on the mRNA stability. So, in the context of lead poisoning, we wanted to see what is the combination of these SNPs on the blood lead levels. And for your convenience, I will be mentioning them as VDR1, VDR2, and VDR3. So here we observed that the homozygous mutant AA of VDR1, the homozygous mutant of TT of VDR2, and the homozygous wild of VDR3 are associated with higher blood lead levels. Since these 3 SNPs are found together in the same chromosome, we did a haplotype analysis, and this is the predicted haplotype from the SHESIS software, and we found that the ATT haplotype combination showed a higher median blood lead level compared to the other haplotype combinations. Finally, we see here the HFE polymorphism. In the HFE polymorphism, with the C to G change, the GG genotype showed a higher median levels of blood lead. Thank you.
Thank you very much. Again, this paper is open for discussion. Any questions, please? Yes.
Is there any correlation in the individuals? Is there any correlation in the individuals having polymorphisms versus the activity of the enzymes?
Yes, sir. Actually, there are literatures which say that this new. Sir, there are literatures which say that with the G to C nucleotide change, the electronegative property of the enzyme changes. So, and that lead binds to the active site of the allied enzyme and it inhibits it. So, what we have in the lead exposed individuals that we have screened, all these they showed higher blood lead levels and corresponding low allied activity.
Any more questions?
What is the biokinetics of clearance of lead from the body?
Sir?
What is the biokinetics of clearance of lead once it gone into the body? Clearance rate.
The half-life is 4 weeks, sir.
4 weeks.
4 weeks.
Any more? Okay, thank you. So we come to the last paper in this session by Miss Suma Prabhu.
A very good afternoon, everyone. I am Suma Prabhu, and I stand before you all today to present a brief gist of our work on comparative toxicological evaluation of bare and pegylated iron oxide nanoparticles for its future clinical applications. Now, before I begin, I would like to start with a small little saying. All great things come in small packages. And why not? Nanoparticles, due to their small size, have actually revolutionized the society and humanity, starting from the technological perspective to the hospitals in clinics for their diagnostic and therapeutic applications. Now, as I say nanoparticles, they can be made up of biological origin, may it be lipids, polymers, or dendrimers, and so on, or it can be organic, such as the metal nanoparticles, including iron, gold, silver, and etc. Among the various available metallic nanoparticles, iron oxide nanoparticles are said to be one of the clinically approved metallic nanoparticle, which is being used as a contrast agent in MRI. And other than that, it provides an enormous surface area for modifications to plantin ligands or antibodies or coating with various substances. Now, as I talk about so many advantages of these nanoparticles, nothing in life comes 100% perfect, and so are these nanoparticles, and so are iron nanoparticles. Now, these are reported to induce enormous toxicity, including generation of oxidative stress, which results in various cellular changes resulting in cell death. Now, keeping all these positive and negative, which is the boon and bane of these nanoparticles, we came up with the objective of the study to develop and synthesize iron oxide nanoparticles and pegylate them, that is, coat them with a polymer called as polyethylene glycol. Now, there are two reasons why we chose to coat these nanoparticles. One, because of its stability to increase its half-life. For that matter, I said we will be further using it for clinical applications. So the half-life in the systemic circulation is of primary importance. And on the other hand, these nanoparticles, as you can see, gets clumped up in the images. So to avoid such aggregation, we had to coat these with polyethylene glycol and did a detailed characterization, which becomes a primary relevance to determine its fate in systemic circulation. And so we did different electron microscopy. And on the other hand, when I say magnetic nanoparticles, we can say children playing around with iron dust on the paper and moving magnet underneath the paper so that it aligns in the movement of the magnet, and so are these magnetic nanoparticle. With the assistance of an expert physicist, we determined that the synthesized iron nanoparticles also retain magnetism and so are called as super paramagnetic iron oxide nanoparticles, which are of immense help for clinical applications. Further, we went on to study the acute toxicity profiling, where we comparatively determine the acute toxicity of iron oxide, which is pegylated and naked iron oxide, which showed an LD50 of 508 mg per kg body weight. Nonetheless, there were no extreme histopathological changes, but we did see deposits of iron in various vital organs. And we selected and shortlisted 1/10 dose of LD50 to further evaluate the genotoxic and other systemic toxicity profile, which did not show any formation of micronucleus at the 1/10 dose, nor was there any formation of, nor were there any chromosomal aberrations which perfectly correlated with the formation of micronucleus. And further, to determine the distribution of all these nanoparticles in the systemic circulation, we harvested the organs and evaluated the total iron content in different vital organs to determine the biodistribution, where we found surprisingly deposits in lungs, liver, and spleen. Further, as reported, there are oxidative stress damage caused due to the exposure of iron oxide nanoparticles. We also evaluated the signature GSH and GST activity, which showed an enhanced profiling in the naked iron oxide exposed mouse 48 hours post intravenous exposure. And further, we also evaluated a detailed serum biochemistry profile to indirectly determine the functionality of organs, where we did not find any significant changes on the 1/10 LD50 dose of systemically exposed mice. And this part of the study has already been published. And in conclusion, I would like to say that a single administrative dose did not show any systemic toxicity. And as I said, we intentionally designed these iron nanoparticles with retained magnetism with an intention of magnetic hyperthermia and multifunctionality. We further developed a iron oxide based nanocomposite to target to the brain tumors, very specifically glioblastomas, which is the second part of the study. These are some of the references. Thank you.
Thank you very much for a nice presentation, and I'm sure this will elicit some questions from the audience.
This pegylation has got one more benefit, that it will prevent the immune reaction. That is one thing. Second thing is that pegylation will not decrease the surface activity of the nanoparticle, because generally they are functionally better because they have got high surface activity.
Yes, sir. So in this study, we used a single peg molecule to enhance the surface functionality. We will be supposingly, like if we intentionally want to tag an antibody or some other moiety in specific and targeting one particular cell type, then we would be, it is suggestive of using a bifunctional peg, so that there are surface ligands where you can chemically bond them and conjugate them.
Okay. Thank you very much.
Yes. Yes, please.
It's not a question, there is a comment like, in all the presentation, we are using bar charts with error bars, but please do mention what error bar stands for. Sometimes we use standard deviation, sometimes we use standard errors. So please do mention in all the graphs what the error bar stands for.
Yes, sir.
It's for all.
Yes, sir. Thank you.
Any more questions?
We use pluronic for coating, surface coating.
Yes,
That
sir.
I think there, you can add, you can add drugs to that.
Yes, sir.
Pegylation, yeah, that is, we found pegylation also we have tried, but in pluronic coated will be, I found it slightly better for adding 1 or 2 drugs to that.
No, because iron oxide, the beauty of these nanoparticles is you can either directly conjugate the drug onto the surface of the particle, or you can either hide the nanoparticles at the center of the molecule and then form a nanocomposite.
Okay, thank you, Ms. Prabhu. Mm. This concludes this session of Young Scientist presentation. And I'm glad that the audience number is very significant, even on the last day. And I'm sure these young scientists got impetus from the presence of, to present their work in front of such an audience. In summarizing the session, I may say that these young scientists, they presented their work on several aspects of toxicology, primarily, including covering heavy metals like lead, cadmium, arsenic, and organic compounds like organophosphorus, pesticides, polycyclic aromatic hydrocarbons, and of course, natural radiation. And lastly, a new area in using nanoparticles of metals. So I thank all of you, all the speakers, and I thank the audience. And last but not the least, on behalf of Dr. Das and myself, I thank the organizers for giving us this opportunity to chair the session. Thank you.
On behalf of the organizers, I thank Dr. Bhattacharya and Dr. Das for chairing the session. Thank you, sir. Thank you, ma'am. I now request Dr. Das to present a memento to Dr. Bhattacharya as a token of our gratitude. Thank you, ma'am. We shall now break for tea. We shall be back in 15 minutes for the concluding session.